Today we are going to continue learning about transgenesis and transgenic organisms. Transgenesis may be defined as the introduction of exogenous DNA into the genome such that it is stably maintained in a heritable manner. Organisms that have been permanently engineered by either gene insertion, gene deletion, or gene replacement are called transgenic organisms, and any foreign or modified genes that are added are called transgenes. A transgenic animal contains a transgene in all of its cells, usually introduced by microinjection of a fertilized egg cell. The first transgenic animal were produced in 1981 by Ralph Prinster and Richard Palmiter. They succeeded in introducing a gene for rat growth hormone (GH) into the fertilized eggs of mice. The injected DNA was constructed so as to contain the coding portion of the rat GH gene in a position just downstream from the promoter region of the mouse metallothionein gene. Transgenic animals are currently being used to study DNA regulatory elements, expression of proteins during differentiation, tissue specificity, and the role of oncogene products on growth, differentiation, and induction of tumorigenesis. Such technologies should permit replacing defective genes in the developing embryo. Mouse transgenesis. Transgenic mice. So transgenes are there. A transgenic animal contains a transgene of all of its cells, usually introduced by microinjection of a fertilized egg cell. A transgenic animal contains a transgene in all of its cells, not only in one cell, in all cells, introduced by microinjection of a fertilized egg cell. The first transgenic animals were produced in 1981 by Ralph Prinster and Richard Palmiter. They succeeded in introducing a gene for rat growth hormone into the fertilized eggs of mice. The injected DNA was constructed so as to contain the coding portion of the rat GH gene in a position just downstream from the promoter region of the mouse metallothionein gene. Transgenic animals are currently being used to study DNA regulation elements, expression of proteins during differentiation, tissue specificity, and the role of oncogene products on growth, differentiation, and introduction of tumorigenesis. Such technology should permit replacing defective genes in developing embryo. They succeed in introduction of a gene of a rat growth hormone into a fertilized egg of mice. The injected DNA was constructed so as to contain the coding portion of the rat GH gene in position just downstream from the promoter region of the mouse metallothionein gene. Transgenic animals are currently being used to study DNA regulatory elements, expression of proteins during differentiation, tissue specificity, and the role of oncogene products on growth, differentiation, and induction of tumorigenesis. Such technology should permit replacing defective genes in developing embryo. Then mouse transgenesis. Transgenic mouse have been produced by injecting DNA into the male pronucleus just after fertilization. Microinjection is done using a microneedle. The microinjected transgene randomly integrates into chromosomal DNA, usually at a single site and usually at multiple copies. It is more common for the DNA to integrate after one or two cell divisions, in which case the resulting mouse is a mosaic containing both transformed and non-transformed cells. The surviving intact eggs after microinjection are implanted into a host female mouse. 3 weeks after birth of the pups, genomic DNA from mouse are analyzed. Mice that score positive by test are then bred to establish founder transgenic lines. Mouse transgenesis. Transgenic mice have been produced by injecting DNA into the male pronucleus just after fertilization. Microinjection is done using a microneedle. The microinjected transgene randomly integrates into chromosomal DNA, usually at a single site and usually as multiple copies. It is more common for DNA to integrate after one or two cell divisions, in which case the resulting mouse is mosaic, containing both transformed and non-transformed cells. The surviving intact eggs after microinjection are implanted into a host female mouse. 3 weeks after birth of the pups, genomic DNA from mouse are analyzed. Mice that score positive by test are then bred to establish founder transgenic lines. Female pronucleus holding pipette, DNA solution, male pronucleus, transfer to oviducts of pseudopregnant female, then transgenic mice. Construction of transgenic mice by pronuclear microinjection. A holding pipette which can hold fertilized oocyte in place, while the microinjection pipette is used to pierce the oocyte and then the male pronucleus. An aqueous solution of the desired DNA is then pipetted directly into the pronucleus. The introduced DNA clones can integrate into chromosomal DNA. Surviving oocytes are re-implanted into the oviducts of pseudopregnant foster female which has been mated with a vasectomized male. The mating act can initiate physiological changes in the male which stimulates the development of the implanted embryos. Gene knockout. A gene knockout or simply knockout is a genetically engineered organism that carries one or more genes in its chromosomes that has been made inoperative. The primary use of this technique has been to replace a normal gene with a mutant allele or a disrupted form of the gene, thus knocking out the gene's function. So, gene knockout or simply knockouts is a genetically engineered organism that carries one or more genes in its chromosome that has been made inoperative. The primary use of this technique has been to replace a normal gene with a mutant allele or a disrupted form of a gene, thus knocking out the gene's functions. Then process of developing knockout mouse. Transgenic mice that carry such a disrupted gene called knockout mice. Production of gene targeted knockout mice involves the following steps: isolation and cutting of embryonic stem cells from the inner cell mass of a mouse blastocyst, then introduction of a mutant. First, isolation and culturing. Isolation and culturing of embryonic stem cells from the inner cell mass of a mouse blastocyst. Introduction of a mutant or disrupted gene into the cultured ES cells and selection of homologous recombinant cells in which the gene of interest has been knocked out, replaced by a non-functional form of the gene. Then injection of homologous recombinant ES cells into a recipient mouse blastocyst and surgical implantation of the blastocyst into a pseudo-pregnant mouse. Mating of chimeric offspring heterozygous for the disrupted gene to produce a homozygous knockout mice. A common approach to inactivate a selected gene is to insert an antibiotic resistance gene known as NEO into the cloned gene of interest. This both inactivates the gene of interest and allows identification of ES cells that have been successfully transfected. The normal gene in a very low percentage of ES cells may have replaced by the neo-disrupted recombinant gene by homologous recombination. So blastocyst is there. Removal of cells of blastocyst and culture of ES cells, then transfection with a cloned mutated non-functional gene, then generation of knockout mouse sequence, surgically transfer embryo into pseudo-pregnant mouse. Transgenic plants. Transgenic plants are those plants which carry additional, stably integrated and expressed foreign genes from trans species. The whole point involving introduction, integration and expression of foreign genes in the host is called genetic transformation. The combined use of recombinant DNA technology, gene transfer methods, and tissue culture techniques has led to the efficient transformation and production of transgenics in a wide variety of crop plants. Unlike conventional breeding, only the cloned genes of agronomic importance is being introduced without the co-transfer and other undesirable gene from the donor. The recipient genotype is least disturbed and there is no need for repeated back crosses. This will serve as an effective means for removing certain specific defects of otherwise adopted cultivars. General procedure used to make a transgenic plant. The first step in the process of development of a transgenic plant involves the formation of the recombinant plasmid and its transfer to plant cells. An agrobacterium plasmid that normally carries the tDNA sequence is modified by substituting a selectable marker such as kanamycin resistance gene and a desired transgene between a 25 nucleotide pair tDNA repeats. Agrobacteria that carrying recombinant plasmid and both a selectable marker and a desired transgene are incubated in culture with plant cells. The wounded cell plant cells at the edge releases substances that attract the agrobacteria and cause them to inject DNA into these cells. Only those plant cells that take up the appropriate DNA and express the selectable marker gene survive to proliferate and form a callus. So the growth factors applied to the callus induces it to form shoots and root and grow into adult plants carrying the transgenic. Transgenic plants application, development of insect, virus and herbicide resistant plant. Bt toxin. Bacillus thuringiensis during sporulation forms intracellular crystalline bodies that contain an insecticidal protein called the delta endotoxin. The delta endotoxin protein, Cry1, Cry2, Cry3, Cry proteins and others that accumulates in the bacterium is an inactive precursor. After ingestion by the insect, this protoxin is cleaved by protease, resulting in shorter versions of protein that display the toxic activity by cleaved by protease, resulting in shorter versions of the protein that display the toxic activity by biting the inside of the insect's gut and damaging the surface epithelium. By the process of genetic engineering, genes for Bacillus thuringiensis toxin can be expressed in plants. Through genetic engineering, it is possible to develop crops that are interestingly resistant to insect. Several different strategies have been used to confer resistance against insect predators. An approach involves a gene for an insecticidal protoxin produced by one of several subspecies of the bacterium Bacillus thuringiensis. Other common strategies use genes for plant proteins such as alpha amylase inhibitors, protease inhibitor, and lectin have been shown to be effective against a wide variety of insects. Development of stress tolerant plant like oxidative stress, salt stress, then modification of plant nutritional content like amino acid, lipid, vitamin, iron, modification of plant nutritional content, and the use of genetic engineering techniques allows scientists to develop the plants with improved characteristics. For example, genetic engineering has been used to improve nutritional quality of several plants by modification of amino acid content of some of their seed storage proteins. For example, rice is extremely low in vitamin A. Portuguese and Bayer developed genetically engineered rice, popularly known as golden rice, which is enriched in provitamin A by introducing three genes involved in the biosynthetic pathway of carotenoids, the precursor for vitamin A. So, the fatty acid composition of both edible and non-edible oil producing crops, the tests of fruits and vegetables by the production of monelin. Plants as bioreactor for production of antibodies, polymers, etc. Plants are easy to grow and can generate considerable biomass. This is the reason the transgenic plants are used for the production of commercial protein and chemicals. Plants has been used to produce monoclonal antibodies and antibody fragments. The polymer polyhydroxyl butyrate, which can be used to make a biodegradable plastic-like material, and a number of potential therapeutic agents like human protein C, anticoagulant, human hirudin variant 2, anticoagulant, human erythropoietin, anemia, human alpha interferon, hepatitis C and B, human growth hormone, dwarfism. Synthesis of edible vaccines. Commercial vaccines are expensive to produce and package and require trained personnel to administer injections. So, would be advantages if vaccines could be delivered inextensively in a broad scale in an edible form, like as part of fruit or vegetable. An edible vaccine, in contrast to traditional vaccines, would not require elaborate production facilities, purification, sterilization, packaging, or specialized delivery system. Modification of plant yield like increased iron and oxygen content. What is molecular farming? It is an application of genetic engineering in which genes, primarily of human or animal origin, are introduced into plants or farm animals for cost-effective production of therapeutic products such as antibodies, blood products, cytokines, growth factors, hormones, recombinant enzymes, and human and veterinary vaccines. Therapeutic compounds so produced are also known as biopharmaceuticals. pharmaceuticals from biological organisms. The organism in which genes coding for target therapeutically active compounds are introduced are often referred to as expression system. Expression system studies so far include bacteria, yeast, plants, viruses, animal cell culture, transgenic plants, and transgenic animals. Initially, bacteria were the most widely used expression system, but due to complexity of the most therapeutic proteins to be produced and simplicity of the bacterial system, new expression systems were explored. As of now, plants are the preferred and most widely used expression system in comparison to other systems. The first recombinant pharmaceutical protein produced in plant was human serum albumin, first produced in 1990 in transgenic tobacco and potato plants. Next, gene therapy. Gene therapy or gene replacement therapy is the insertion of genes into an individual's cells and tissues to treat a disease and hereditary disease in particular. Gene therapy typically aims to supplement a defective mutant allele in a with a functional one. Gene therapy may be germline or somatic cell gene therapy. Current gene therapy is exclusively somatic gene therapy, the introduction of genes into somatic cells of an affected individual. Germline cell therapy involves the permanent transmission modification of genome of a gamete, a zygote, or an early embryo. The prospect of human germline gene theory is currently not sanctioned. Gene therapy may be classical or non-classical gene therapy. In classical gene therapy, genes are delivered to appropriate target cells with the aim of obtaining optimal expression of the introduced genes. The idea of non-classical gene therapy is to inhibit the expression of gene associated with the pathogenesis or to correct a genetic defect and to restore normal gene expression. Methods for inserting and expressing a gene in a target cell. The genetic material may be transferred directly into cells within a patient in vivo gene therapy, or cells may be removed from the patient and the genetic material inserted into them in vivo prior to transplanting the modified cells back into the patient. Ex vivo gene therapy. Ex vivo gene transfer, this initially involves transfer of cloned genes into cells grown in culture. Those cells which have been transformed successfully are selected, expanded by cell culture in vitro, and then introduced into the patient. This approach is only applicable to tissues that can be removed from the body, altered genetically, and returned to the patient where they will engraft and survive for a long period of time. Cells of the hematopoietic system and skin cells, for example. Then in vivo gene transfer. The cloned genes are transferred directly into the tissues of the patient. This may be the only possible option in tissues where individual cells cannot be cultured in vitro in sufficient numbers. The cloned genes are transferred directly to the tissues of the patient. For example, brain cells and our culture cells cannot be reimplanted efficiently in patients. In vivo, cells removed, patient cell, gene transfer, same cells X plus return genetically modified cells to patient. The in vivo and ex vivo gene therapy. Gene delivery system. One of the biggest problems in somatic gene therapy is to find an effective gene delivery system. Two basic strategies have been investigated, like viral based vectors and non-viral transfection methods. There are currently two non-viral transfection methods like liposome and biolistics being tested for in vivo and ex vivo gene therapy approaches. Viral based vectors, viruses attack their hosts to insert their genetic material into the genetic material of the host. Continuation we will be learning after the uploading of next video.